The overall goal of the following experiment is to use electric cell substrate impedance sensing, known as ESIS, to characterize cellular behaviors. This is achieved by growing cells on electrodes, in an array, and taking measurements in different modes to quantify endothelial barrier formation, maturation, and function. Manipulations such as electrical wounding and adding stimulants are then made to test cell motility and barrier function. Results are obtained that describe cell attachment, proliferation, migration, detection of endothelial permeability, and assessment of cell-cell and cell-substrate contacts based on ESIS.
This method can help to answer key questions in the cell biological field, such as providing online generation of quantitative data characterizing cells in their natural confluent state under standard cell culture conditions.
Generally, individuals new to this method will struggle because the underlying theory appears complex, and you need to be aware of several basic considerations before you start an experiment.
To begin, the arrays must be cleaned and stabilized to prevent electrode drift, improve reproducibility, and increase the signal strength ratio. Therefore, add 200 microliters of 10 millimolar L-cysteine to each well of an 8-well array. After 15 minutes at room temperature, remove the L-cysteine with 2 ultra-pure water washes, not phosphate buffers. Also, do not expose the electrodes to serum-containing solutions before coating, as they can interfere with protein.